Hello students. Earlier in the last video we have discussed about the 2 points about basis of classification. The first basis we have discussed about the level of organization. That means their body is made up of the cells, that is cellular level of organization, where the cells were only aggregated, loosely aggregated, but they actually do not form any kind of aggregation, and that was not forming any tissue. Later on, the cells were aggregated. Whenever evolution took place, cells were starting to aggregate. Then after that, they start to become specialized to perform some function that is known as the tissue grade of organization. And gradually, those tissues start to unite. They start to perform some another functions, become more and more specialized. That was known as organ level of organization. And the last level of organization we discussed was the organ system level of organization. Hope you have understood all these things. The different types of organisms, the phylums we have discussed. If it is the cellular level of organization, then the sponges, the poriferans, they have the cellular level of organization. If it is tissue, then the coelentrates and the ctenophores, they come under the tissue level of organization. Then the organ level of organization we have seen in case of the platyhelminths, and after that we have seen the organ system level of organization that was found from the Escalmentis up to the last phylum, it is Chordata. Then after that we have discussed the next level of next basis of classification, that was symmetry. So in that symmetry we have seen actually the asymmetrical organism, where there is no plane is possible. If you make suppose a axis passing through the midpoint of that organism, then also you are not able to get the two equal halves. So what are those? These are the asymmetrical organism. I told you that in case of Phylum Porifera, except Calcarea, this is a class of the Phylum Porifera. So what was that? That phylum actually, the Phylum Porifera comes under the asymmetrical organism. Next one we have seen, these are the radially symmetrical organism, where we can have numbers of horizontal planes also, vertical planes also. You can get numbers of planes, and after that we can able to get two equal halves. So that was the second basis, that is the radial symmetry. We have seen another organism too, that was starfish. You can see the starfishes, they have 5 arms only. So that is also coming under the radially symmetrical organism. But that radially symmetrical organism, that is Echinodermata, they have two forms. First of all, need to know that the larva and the adult. As we already know that what is larva and what is adult. If you see a tadpole, that is a larva, but that tadpole become adult and become a frog that we see normally. So that frog is the adult. So similarly, what happened in case of echinoderms, the echinoderms are only the adult one. The adult echinoderms are only radially symmetrical organism. After that, we have seen the bilaterally symmetrical animals, which are including all those animals. We are also included here. What happened to those animals? That if you have one plane only, two planes it is possible, two identical halves we can able to get. Only passing through that midpoint, only one axis is possible. Either it can be horizontal or it can be vertical. So what is that? That is the bilaterally symmetrical animal. What happened to the bilaterally symmetrical animal? We will get two equal halves, but only one axis is possible. I told that in the radially symmetrical animal, the echinoderms, those echinoderms which are larval stages. The larval stages are actually bilaterally symmetrical animal, but in the same echinoderms, the adult one, that is actually radially symmetrical animal. So another one, the other organisms like platyhelminths, escalmentis, we can see annelids, arthropoda, mollusca, exabechinoderm, then hemichordata and chordata, they are also coming under the bilaterally symmetrical animal. We will see a number of exceptions too. So after that, the next basis of classification, let's just see. The next basis of classification, the third basis of classification is the germ layer. Okay. So now just see what is germ layer. Germ layer is germinal layer. Don't think about it's the pathogen or some germs or like that. No, it's the germinal layer. What happens? See, our life is started from a single cell state. So what happened? First of all, from the male, a spermatozoa, a sperm that comes, and from the mother, a cell, that is a ovum, comes. So that sperm and the ovum, it fertilize, actually the mixing of the gametes, that is called fertilization. So in class 8 already you have studied what is fertilization, what is mitosis, what is meiosis, all these things you have studied. So we will see in detail. So those points, they are a little bit will be required here also to understand all these things. So what happened here? The sperm and the ovum, they fuse or they fertilize to form a zygote. So that zygote undergo mitosis, and after that it form a number of cells. Now just see what happens whenever all this mitosis processes are going on. At that time what happened? Gradually the cells start to undergo a actually arrangement, and whenever all the cells are started to arrange, a solid layer has been formed, and that solid layer, one is formed, then after that the another solid layer will be formed, that will be made up of cells only. So this way the solid layer started to form, and from those solid layers different type of organs are being started to form. So what is happening here? See, in case of germinal layer, first of all, from, suppose this is the zygote, it undergo first mitosis, it produce 2 cell, 2 to 4 cell. So gradually this way the cells are started to form. What happened at one period of time, some cells are formed, and those cells, suppose they started to linearly present. It is actually primitive string. We will understand it later whenever we will get in a second year embryogenesis. We'll get it, all these things. So just remember here now that from the zygote, whenever it undergo mitosis, then a solid cell layer is been formed. So that solid cell layer is known as endoderm. Then after that another cell layer will be formed above this, another layer that is known as ectoderm. So this way also another cell layer is been formed. This is known as ectoderm. And gradually what happened, some of the cells, they started to present in between, but those cells are not forming any lining, or they are not even trying, they're trying to form, but they're not able to completely get formed. So this way the germinal layers are formed. See, first of all, in case of germinal layer, we will get two types of animals. First animal, they are diploblastic. So what is diploblastic animal? And another type of animals on this germ layer basis, they are triploblastic. So just see what is diploblastic and triploblastic animal. First of all, in case of those diploblastic animal, for the first time, one germinal layer has been formed, that is endoderm. Suppose the first germinal layer has been formed, that is endoderm. Then after that, the another germinal layer is also formed, that is ectoderm. But what happened, that in between the endoderm and ectoderm, there is no cell, or there is no complete cell layer, or any unite or aggregation of cells are not been able to form till now. What is that? So there is a space developed, and in that space only a jelly-like matrix will be found. It is not at all a tissue layer. It is just a jelly-like matrix. So what happened in case of those animals which have only outer, this outer ectoderm is there. This is the outer ectoderm, and inside they are going to have inner endoderm, but in between the ectoderm and the endoderm there is no cell layer is been formed in the embryonic state. So what happened, that jelly like matrix, what we can call it, this is the mesoglia. So that type of organism which are not able to form the middle layer, or in the in between the ectoderm and in between the ectoderm and the endoderm, no germinal layer has been found, just a non-cellular jelly like matrix will be there, that will be known as diploblastic animal. So what happened to the diploblastic animal in their embryonic state? Whenever this embryo is getting formed, actually they will have only two germinal layers, but later on all these germinal layers will form different organs, and after that the complete organism will be formed. But in the embryonic state, whenever the germinal layers are been formed, only two germinal layers are possible. So whenever this organism is been formed in the embryonic state, only two germinal layers are there, only ectoderm and endoderm, and in between them there is a jelly-like matrix has been developed. So this is known as mesoglia. That type of animals, they are known as diploblastic animal. Now just see the next organism. See, already outer ectoderm is already present. So this is ectoderm, this is already present. Inner endoderm, which is earlier formed. So ectoderm and endoderm are already there. Outer ectoderm, inner endoderm, and in between, instead of jelly-like matrix, see what happened whenever evolution took place. So that jelly-like matrix get thicker and thicker and thicker. The cells started to form, the cells started to get reorganized, and after that it formed a layer of cells. So later on, whenever evolution took place, then in those animals they also start to form the third germinal layer in between ectoderm and the endoderm. So this is known as now a solid layer of cell. This is known as mesoderm. So those animal where in the embryonic state they form ectoderm and endoderm, along with that in between ectoderm and endoderm they also can form the third germinal layer, that is mesoderm. That type of animals, they are known as triploblastic animal. Now just see the organisms which will come under diploblastic and triploblastic. Obviously evolution will be started from simplest organism to get complex. So diploblastic organisms are the simple one. Then after that evolution took place and they become more and more complex and become triploblastic. Just see the first, I told the first phylum. What was the first phylum? This is Phylum Porifera. Then after that Coelenterata. Then after that Ctenophora. Then after that we will have platyhelminths. Then gradually we'll have all these other animals like Escalminths, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. So what happened in case of these animals? See, what happened to the phylum Porifera? I told in the earlier basis of classification in the level of organization that in case of the animals Porifera, they will have only cells. So this type of animal are not even capable of forming even any of the germinal layers. Their embryonic state will also have only cells. So their body is made up of only cells. They are not able to form any of the germinal layers. They will have germinal layer type of layers. So those layers will be constituting the body wall. They will have outer cell layer, inner cell layer, in between they will have a jelly-like matrix, but this is not a germ layer. So what happened in case of Phylum Porifera? They will not have any type of germinal layer. So they will have actually outer pinacoderm and inner choanoderm. Will discuss in detail whenever we'll see all those phylums later, but just remember that till now the Phylum Porifera, they are not even become diploblastic animal. They will have only some aggregation of cells. So they will have two layers, outer layer and inner layer. So outer layer is known as pinacoderm and inner layer is known as the choanoderm. So that is the Phylum Porifera, which do not have any germinal layer. Now after that come to the diploblastic animal. Whenever it come to the diploblastic animal, they are going to have the outer, this is outer ectoderm, inner endoderm is present, and in between ectoderm and inner endoderm they will have a jelly-like matrix which is very much liquid or jelly-like matrix. So what is that? This is known as mesoglia. So what type of animal it is going to have? these are the phylum Coelentrates, ctenophores, they are diploblastic. Now just see, after that we will have the last type of basis, that is triploblastic animal. So what happened whenever evolution took place, that mesoglia become more and more thicker, this cells started to form, they developed the germinal layers in the embryonic state. So what happened now, those cells are started to form in between ectoderm and endoderm. So the last germinal layer which is formed, that is in between ectoderm and endoderm, that is known as mesoderm. So those animals which can now form the ectoderm, endoderm as well as the mesoderm, that type of animals are known as triploblastic. So what type of animals are triploblastic animals then? See, the phylum Platyhelminths, after the Platyhelminths up to the Chordata, without exception, all of the animals, all of the phylums, they are going to be triploblastic animals. So from Platyhelminths up to Chordata, all of the animals, they are triploblastic. So that is the third basis of classification, that is germinal layer. We'll have a group of animal, Phylum Porifera, which is not able to form any germinal layer. Second group of animal which are diploblastic, which have two germinal layers only, ectoderm and endoderm. In between they will have just jelly-like matrix, mesoglia. So Phylum Coelentrata and Ctenophora, they are going to have diploblastic organization, and after that they will have triploblastic. That means now evolution took place, and after that in that jelly-like matrix become more and more condensed, and after that the cells started to form. So that cells, they are known as the mesoderm in between ectoderm and endoderm. So they are known as the triploblastic animals. So the Platyhelminths up to chordates, all of the animals are triploblastic. Now just see the next basis of classification. Now the next basis of classification, see, the fourth basis of classification is the coelom. Now what is coelom? Coelom is actually the body cavity. Now what is body cavity or what is coelom? Coelom is actually one structure which is present in between the outer body wall and the inner gut wall. See, the alimentary canal is also called as the gut. So that gut wall, suppose this is the alimentary canal and this is the outer body wall. So this is the body wall and this is the gut wall. And what is coelom now? This is the whole gut, so this will be the gut wall. So what happened in between the gut wall and the body wall, we have some cavities, and those cavities are known as coelom. One more thing you should know, that this body cavity, that coelom, that is actually formed from one germinal layer, that is mesoderm. So what happened? This coelom, this is actually developed from the mesoderm. This is developed from the mesoderm. And the second thing, remember that this coelom, that is actually present in between. This is present in between the body wall and the gut wall. So what is that? This is coelom. Remember, the coelom will be formed from the mesoderm, which is present in between the gut, that is the lining of the alimentary canal, and the outer body wall. Now just see how many types of animals you can see on the basis of this coelom. First of all, you are going to have, see, depending upon the presence or absence or coelom, we are going to have the first type of animal. They are acoelomate. Then after that you have Pseudo Selamat. Then after that you are going to have truly coelomate or eucoelomate, or simply you can call it also coelomate. So just see what are those animals acoelomate. So obviously those organisms which do not have mesoderm, how can they will form the coelom? Remember, the coelom is formed from the mesoderm. So those organisms which do not have even the mesoderm, that is a diploblastic animals, so those diploblastic animals will be a acoelomate animal. And remember the first triploblastic animal? The first triploblastic animal was platyhelminths. So what type of animal it is? See, in case of platyhelminths, they actually develop this mesoderm. But this mesoderm is not able to, it is not capable enough to form the coelom or the body cavity. So all of the diploblastic animal, that is not even Porifera. Porifera is not going to have even the germ layer. So Porifera, coelentrates, ctenophores, platyhelminths. So this type of animals, they do not have the either they do not have the mesoderm, diploblastic animal, or if they have the mesoderm, like in case of phylum platyhelminths, they have the mesoderm, but this mesoderm is not able to form the germinal, they are not able to form the coelom. So that type of animal, they are the acoelomate animal. Just take the example, this is the outer, suppose, ectoderm, this is the innermost endoderm. Okay, so in between them, if suppose there is no germinal layer, it is just the mesoglia, then what will happen? Is it possible that they can form the coelom? No. So this type of animals, they are acoelomate type of animal. So just see what are those: phylum Porifera, which do not have any germinal layer. Next one, that is Coelenterata, Ctenophora. Then after that, the animal which have the germinal layer, but that mesoderm is not able to form the coelom, that is platyhelminths. So this animal is also acoelomate animal. So which have the in between the ectoderm and endoderm, there is no cavity present. They are not even going to form this cavity due to the absence of the mesoderm, or due to the incapability of this form of this mesoderm to form this coelom or body cavity. They are acoelomate animal. Next one, see pseudo Selamat animal. What is happening now? So in case of pseudo Selamat animal, they will have the outer ectoderm. So outer ectoderm is present, and now they have this endoderm too. But what is happening? This mesoglia, which is present in case of diploblastic animal, they start to become more and more thicker, and after that it forms the cells. But if this mesoderm is like they're splitting up, they're forming some pouch-like structure, suppose this is mesoderm, then what you are going to call it? This mesoderm is being formed only, but this mesoderm is not able to form the coelom properly. So this coelom is present as some pouches, isn't that? So what is that? This is the pseudo Selamat animal. They will have the ectoderm, they will have the endoderm, and this mesoderm that is actually formed as just splitting up of mesoderm. So this is the mesoderm, which is forming some pouch-like structure in between ectoderm and endoderm. So that's why, so this type of animal, this animal which have not a true cavity, so this is known as the pseudo Selamat animal, which is going to have some pouch only. So this is pseudo coelom. One more thing, just remember that the coelom, to be called as a true coelom, they need to have another criteria too, that the coelom should be lined. The coelom should be lined by mesoderm. So from all the sides, that coelom should be lined by mesoderm, then only we can call it as the true coelom. Now here you can see, in case of that coelom, you can see this coelom, suppose here this is the coelom. So above they are going to have ectoderm, here they are going to have endoderm. How can we call it as the truly a Selamat animal? So this type of organism where we can able to see the coelom, the body cavity is there. In between body wall and the gut wall, they are going to have the coelom, but this coelom is not a true coelom. Why? Because it is not forming, it is not lined by the mesoderm only. So that's why this type of animal, they are known as the pseudo Selamat animal. So how many types of animals there are pseudo Selamat? One only phylum. Remember, these are the Escalmintis. So in case of Escalmintis, this type of animals, they are known as the Pseudo Selamat animal. Now the last one, that is Selamat animal. Just see what is Selamat animal. Now they are going to have the outer ectoderm. They are going to have endoderm. Now just see, Mesoderm is there, that is completely been developed, and this cavity, see, this is the Mesoderm, here this is the Mesoderm, and in between the Mesoderm they are forming the cavity. See, this is the cavity, this is called as true coelom, where the coelom from all the sides, it is lined by the Mesoderm only. That type of animals, they are known as the coelomate animal. So how many types of animals are included here? Right from the Annelida up to the Chordata. Okay, so from the annelids up to the chordates, they are coelomate animal. Now under that also we are going to have two more division. That means in the coelomate organisms also we are going to have two types of animals. What are they? One is Pseudo Selamat animal, another is the Entero Selamat animal. So what is that? So in case of Pseudo Selamat animal, you can see that in case of Pseudo Selamat animal, they are going to have this Mesoderm. This Mesoderm is derived from splitting up of the gut. So from the gut, as it is formed, but it is splitting up of split it up, and this Mesoderm itself is just like some splitting up structure. So that type of organism are Pseudo Selamat animal. So how many types of animals are included in the Pseudo Selamat animal? See. So another one also, see, Entero Selamat. What is Entero Selamat? Where the Mesoderm is developed from the embryonic gut. That type of animals, they are known as the Enteroselamat animal. So what is that? This is two type of Selamat animal itself. One is Pseudo-Selamat, another is Enteroselamat. So what are those? See, first of all, Annelida, Arthropoda, Mollusca. So this type of animals, they are Pseudo-Selamat. Then after that, in case of Enteroselamat, you can have Echinodermata up to the Chordata. This type of animals, they are Enteroselamat animal. So these are the three types of organisms. Remember, only the different organism which are included in which type of category, that is important. Only see, if it is coelom, that coelom should be lined by the Mesoderm. The Mesoderm will only form the coelom. So that coelom will be covered by, lined by the Mesoderm always. Then next one, this lining, that body cavity, that will be present in between the body wall and the gut wall. Then only we can call it as the coelom. Now on the basis of the coelom, we are going to have three types of animals. First of all, Acoelomate, which do not have this Mesoderm, or they have the Mesoderm but they are not able to form the organism that is Acoelomate, so which do not have the body cavity. Phylum Porifera, Coelenterata, Ctenophora and Platyhelminths, they are Acoelomate. Next one, Pseudo Selamat. What are they? They have the Mesoderm, but these Mesoderms are present as some scattered pouches. So that's why you can see the pseudocoelom here. These are the pseudocoelom, but that coelom is not lined by the Mesoderm always. So that's why that type of animal are known as Pseudo-Selamat animal. What is that phylum? Escalminths. This Escalminths, this animal, this is a phylum which is Pseudo Selamat. Now last one, that is the Euselamat animal or Selamat animal, which have the mesoderm. They form the body cavity, coelom is there, coelom is lined by the mesoderm. So that type of animal are Selamat animal. So coelom has been developed here, so coelom has been formed. Now under that Selamat animal itself, we are going to have two categories. One is Pseudo Selamat, where the mesoderm is developed from the splitting up of structure, and the enterocoelom, which actually derived from the embryonic gut or alimentary canal. They are known as the enterocoelomate animal. So what are they? We are going to have Annelida, Arthropoda and Mollusca. They are Pseudocoelomate animal. And then after that enterocoelomate, which is from the Echinodermata, we will have Hemichordata and the chordata. They are coming under the enterocoelomate animal. So these are the two bases of classification we have seen. In the next video, we'll see the next another basis of classification. Thank you.